Pleasure to welcome back Pam Reinagle to UC Berkeley, and I've known Pam for a long time, probably 20 years or so, since she instigated this movement, which is the National Natural Scene Statistics meeting. I'll just turn off the lights to show all the handsome figures in this photograph. So this was kind of a nascent effort that started in the mid-1990s. So now we hear people talk about natural scene statistics and so forth, and it's just kind of part of the vocabulary. Back in the mid-90s, that wasn't part of the vocabulary of the field. It was kind of a very avant-garde idea. And there was just a few people working on this stuff, and so Pam got this bee in her bonnet that it would be good to get people together who are thinking about these ideas and talk together, about what's going on. They're coming from different fields, like David Donahoe from statistics, and Irisa Mincelli from vision science, Jack Gallant, lots of people from neurophysiology, math, statistics, and so forth. And it was really the beginning of something that really continued for another decade or so, and Pam then launched this into the Gordon Conference, which continued for another maybe 8 or so years. So that was really a fabulous period. And so her interests have turned to other studies in decision making, and so she has a lab at UC San Diego where she's studying decision making in rats and sometimes mice, and so it's that part she's going to be telling us more about today. So thanks a lot for coming.
Great. Thank you for that introduction and that.
Oops, oh yeah, I got it.
Oh yeah, I'm sorry, that's not you. I need my signal back.
Yes, right.
Where'd it go?
And then we just,
I don't know where that cable went.
All right, there we go.
I'm going to turn
And I will
this. Let's see if this is. Oh, this was working before.
now I ruined it.
Yeah. Now I have to reset. There we go. Okay.
Right.
So thanks for coming out for my talk and for inviting me. And today I'm going to talk to you about one of the projects in my lab, which has to do with decision making. And with apologies to Steinbeck in the title here, I'm just going to talk about some research, experimental research and modeling that we've been doing to try and understand why the decision making of rodents and of primates is different and what about it is the same. Okay, so just to orient you to the question that we're interested in decision making, this is an example of a couple of primates. They're foraging here for persimmons. And the decision that most people think about first is there's a lot of different persimmons here, and they have to make a decision which is the best one to harvest. And that's part of what it is we're studying. But the other part is that there's a period of information gathering where they're using their senses to get evidence about the quality of, let's say, the different targets. And then there's a moment at which they decide to commit to one decision or another, right? So this individual has already committed to a decision and is reaching for some persimmon. This individual is still taking in sensory evidence and hasn't yet reached that moment when they've decided to decide. So these two things interact, both what you choose and when you choose it, when you're making a sensory decision like this. And it turns out that this is a problem that's not only solved by humans, but other primates also have to forage for persimmons, and so do rats. And so, so today I'm going to talk about some work we've done trying to compare data from the literature in primates, previous data from human subjects, as well as some of our own new data and data that we've collected from rats in a decision-making task in which there's an open-ended period of time where the subject gets to survey sensory evidence and, when ready, commit to a decision. Okay, so the outline of the talk, just to keep you oriented and keep me on schedule, I'm going to start by just telling you the initial experimental observation. I think maybe a laser pointer would work better, in fact. Yeah, so I'll start with just very briefly the initial experimental observation that led my lab down this path to study this problem. And then I'm going to take a significant number of slides, like maybe 12 slides, reviewing the properties of drift diffusion models of decision making. For those of you who are aficionados in decision making, this is going to be very basic review, but it's important that everybody go over it in some detail because the distinctions that I'm going to be making in the data depend on having a few things straight. And then I'm going to get to the meat of the talk, which is an experimental section where I'm going to talk about new data that we've collected in rats and in human subjects to try and check whether the initial conundrum is experimentally a true fact, and then modeling to try and make sense out of how we can understand both rodent and human or primate decision making in a common framework. So we studied decision making in lots of different visual tasks, but everything I'm going to talk about today was going to use this task here. So you're looking at the back end of a rat who's facing a computer monitor. There are sensors to the left and right that the rat can lick to indicate a choice, and if he makes the correct choice, then out of that location at that moment will come a drop of water. If he makes the wrong choice, then there'll be a timeout when he can't perform any more tasks in a second or two before he can begin another task. And there's one you can't see in the center, and when the rat triggers that sensor, he doesn't get any reward, but that's what makes the visual display begin. So whenever the rat wants to do a trial, they request a trial by licking that sensor. A stimulus comes up on the screen, and these dots, which are randomly located on the screen, will start moving. And this is a random dot motion task, so some of those dots are going to be moving coherently either to the left or the right, and here about half of the dots are moving to the left, as you can see by those little arrows. But other dots are moving at random, and that creates a noise. So there's a signal to noise problem that the animal has to solve to discriminate which is the net motion flow to the left or to the right. There's only two possibilities, so it's a 2AFC task. And that visual stimulus will persist for as long as the animal cares to stare at it until it makes a decision. So now that you're oriented, I'll just show you a few trials. And I chose a very high coherence where the motion is quite obvious so that it's easy for you to tell whether the rat's getting it right. He starts the movie, makes a choice, and he's getting water. I think he gets every single one right in this movie. So this is just qualitatively what the task looks like while the animal's doing it. And so again, he's in control of both which of the two choices he makes and when he commits to that decision. And we set up this experiment in order to try to replicate a very well-established result from the primate and human psychophysics literature. And that is shown in this graph, which I took from Palmer, Hook, and Shadlin's 2005 paper, which was human subjects. And these are just two different subjects. And on each point is the result from a single experiment. And in that experiment, we've plotted the average reaction time of all the correct responses versus the average reaction time of all the errors. Okay? And what if errors and correct choices were roughly the same speed, they should all fall in the diagonal. But what they observed is that on the whole, the error trials tend to be slightly longer. So in the trials that the response was a little late, it's more likely to be a mistake. And that was true for these two subjects. It had been previously reported to be true for macaque monkeys, but they don't have a graph like this that I can show you. So, and it's been replicated in many, many experiments in many labs with both humans and monkeys. So this fact that when primates make an open-ended decision, a freely timed choice like this task, they tend to make more mistakes in those trials that they responded later. So I'll explain to you in the DDM orientation part why that's interesting from a theoretical point of view. But there had been a considerable amount of theoretical interest in trying to explain this fact. And a bunch of different models were being developed, competing models that could explain this fact. And so we thought, why don't we set up a rat model of this with the hope that once we replicated this result, we would go forward to be able to use the rodent model to try to explain the underlying mechanisms. But to our surprise, we did not replicate the result, and in fact we got the opposite result. So this is data from our first paper on this in 2013 from rats. And here the correct reaction time, just like the primates, there's a huge difference in different experimental conditions in different subjects. Any given symbol could be very fast reaction times or very slow reaction times, but within an experiment, the correct reaction times always tended to be the longer ones. So below the diagonal means that when they took more time before they responded, they're more likely to get it correct. So some other ways to look at this, which are not usually plotted in the primate literature, but I find instructive. So this is just the same fact telling you in a different way. Here I'm taking all the trials that a rat did in one experiment and I'm breaking them down by reaction time, and I'm asking what fraction of the trials were correct that had any given reaction time. And in those trials that had very fast reaction times, the rat is barely above chance, 50% correct in classifying that motion stimulus. In the trials where the rat didn't respond for 2 seconds, they were almost 100% correct. So there's an increase in accuracy as a function of reaction time in the task. And yet a third way to visualize this would be to take the reaction time distribution of the animal in an experiment and just pull out the fastest quartile of all its responses or the slowest quartile of all its responses and ask what were the accuracy of their slowest responses or fastest responses. So it's taking the two ends of this distribution and it takes care of the fact that like these reaction times out here are actually extremely rare. So we want to have a sort of equal number. And again, here the percent correct of the slow responses is systematically much higher than the percent correct of the fast responses in the same experiment. So they're all lying above that diagonal. So this was our conundrum. We seem to have a difference between primates and rats. And who would have thought that primate and rodent decision making would be different? Our prior was that it should be highly conserved, at least at the level of these very rapid sensory decisions. Okay, so that's our initial conundrum. For humans and monkeys, decision accuracy declines with reaction time, whereas for rats, decision accuracy increases with reaction time. And now I'm going to spend some time talking about drift diffusion models of decision making to explain why not only the fact that humans and rats are different is interesting, but each of these results individually is surprising. Okay, so this is not the model that we're going to use for our modeling. The model that I'll show you at the end is a completely analytical model. This is just a toy model simulation to introduce the idea of drift diffusion. So suppose that we have a noisy visual motion signal like I showed you, and that there's a very weak bias towards rightward motion, and you find 16 cells somewhere in the brain that are tuned to rightward motion, and I've simulated the stupidest possible neurons. They're just Poisson, completely independent Poisson spike trains that have some firing rate that represents the strength of rightward motion in this complex motion signal. And imagine that we found 16 other neurons that are tuned to leftward motion. They're also getting stimulated because there's also things moving to the left in the display, and their firing rate's just a little bit lower. So there's a difference in firing rate, and that's the evidence the animal is going to have to use to decide, is this motion to the right or to the left? And here I'm just showing if you took the average firing rate of these neurons in any short time window, I think it's maybe a 10 millisecond time window, it's pretty hard to tell that on average, the green, or correct direction of motion, has a higher firing rate than below. You would need to somehow integrate information over time to pull that signal out of the noise. Okay, so there's those instantaneous firing rates of our toy neurons shown at the right. And the idea of the drift diffusion is that somewhere in the brain there's an evidence accumulator. And again, this is just the simplest, stupidest evidence accumulator in the toy model. We're going to imagine somewhere is a neuron that's counting up how many spikes came from the neurons encoding the correct direction, subtract from that the number of spikes that came from neurons encoding the incorrect direction, and just keep counting until it's time you've decided to make a decision. And if you do this, you end up with a time series that looks like this. So it's very noisy. You sometimes get more spikes for the correct direction. Sometimes you get more spikes for the error direction, but you're adding them up over time. And so if there is truly a bias or a signal, there'll be a drift in this random walk in the direction of the correct response, which by convention I'm going to show as being upward in all these plots. So the average drift of this process depends on how strong that motion signal is, or how much stronger is the evidence for right versus left. If you, to illustrate that, here I've actually taken the average of thousands of such trajectories from such a simulation. So in the example I showed, where there's a very weak signal, on average the trajectory slopes up. If there were no difference between the two firing rates, then on average it goes nowhere. And if you had a very strong visual signal, so one firing rate was much stronger than the other, then it would slope up more steeply. And the idea of the drift diffusion model is that the organism has decided that it's going to be satisfied with some particular amount of evidence. Let's say it wants to have 2 arbitrary units of evidence before it makes a decision, and it's just going to wait for this accumulated evidence to hit that threshold, and then whenever a threshold is hit, it's going to make a decision. So here's what happens if you implement that. We have this weak motion stimulus with a very small difference in firing rates, and I've assumed that the animal begins with a neutral prior that assumes it could be either right or left, which would be accurate in our experiment. And then it begins accumulating, and I've just shown the first 8 individual random walk traces that happened to cross the correct threshold first in green traces, and the first 8 that happened to cross the wrong threshold first in red traces. So you can see that it's incredibly variable from trial to trial. There's a lot of noise in what's going to happen in an individual trial. Nevertheless, perhaps surprisingly, this simple decision rule makes the correct choice and recognizes that the firing rate of the rightward coding neurons is higher in 77% of trials. This is a surprisingly efficient algorithm for getting the right answer. And it, on average, takes about 1.5 seconds for it to converge on one or the other answer. So it's also efficient in terms of it doesn't take that much time. The statistics underneath why this is such a good algorithm was first worked out by Wald in 1945, the sequential sampling algorithm, and was first applied to perceptual decision making by Stone in 1960. So this is old news. And again, just to illustrate the properties of the model, I'm going to point out that if we had a stronger motion stimulus, what that means is a bigger difference in firing rates, then these trajectories will tend to cross a threshold sooner, because evidence is accumulating more rapidly. That average slope is increased. And so before we had a difference in firing rate 40 versus 39 hertz, and it was correct 77% of the time. Now it's a bigger difference, 41 versus 38 hertz, between the correct and incorrect directions, and it's going to get to the correct decision 97% of the time. So if you make the visual signal stronger, then a decision rule that's based on this is going to reach the correct response more often. And it's also going to reach its decision faster. So it's going to commit to one or the other decision in less time. And as you can imagine, if I made the stimulus extremely weak, actually this is no motion stimulus at all, so this is pure noise. These trajectories do still hit a threshold. They doesn't just sit there and perseverate indefinitely. It reaches a decision. That decision is right or left 50-50 because there is no right answer. And it takes longer, but not hugely longer, right? It doesn't sit there all day. It comes to some decision in a reasonably short amount of time. But the trend that you can see is the stronger the motion signal, the more accurate the decision would be, and the faster the decision would be. Okay, so this is how the drift diffusion model explains the experimental observation of psychometric curves and chronometric curves. So here's a psychometric curve in this toy model. We have some firing rate difference, which is our signal strength. We have the accuracy of the decision that's reached by this simulation. And it goes from chance when there is no signal and climbs to about 100% when the signal is very strong. And here is the mean reaction time of that model as a function of stimulus strength, and you see that as the stimuli get stronger and stronger, the time required to come to a conclusion is shorter and shorter. So this is the first property of the drift diffusion model that makes it an appealing model for the experimental data we have on decision making, which is that as signal strength increases, decisions become more accurate and faster. And as a result of this, in this sense, accuracy is negatively correlated with reaction time, right? Okay, now I'm going to talk about a second property of that model, which has to do with the threshold. So I'm going to go back to that weak motion stimulus that had 40 versus 39 hertz, so a weak motion stimulus. And before we looked at the decisions that were reached if the threshold was set to, I believe it is 3, which is shown by these dashed lines. And compare that to what happens if I just make the threshold higher. So I'm going to require more information before I reach a decision. I'm going to be a more conservative decision maker. So first of all, on average, it takes a little longer for a trajectory to reach that threshold. So your decisions slow down, right? Again, these are just truly chosen at random the first 8 trajectories that crossed the correct and the first 8 trajectories that crossed the error. This one did eventually cross the error threshold, which is why it's colored red, although it didn't happen yet in 5 seconds. Okay, so what happens when you make the threshold larger, we go from 3 to 3.5, is that more decisions are correct. So you've waited for more evidence. And so sort of by central limit theorem, you're more likely to come up with the correct answer. And so your accuracy increases. And it takes you more time to make a decision, so your reaction time also increases. And so that explains another well-established experimental fact in the literature, which is the speed-accuracy trade-off. If you tell a subject to hurry up, they'll go faster and make more mistakes. If you tell a subject, you better not make a mistake, they slow down and become more accurate. So here, as the threshold or decision criterion gets stronger and stronger, the accuracy of this toy model gets better and better. Concomitant with that, the reaction time of its decisions gets longer and longer. So this is a second property of drift diffusion models. As your threshold or decision criterion increases or becomes more conservative, your decisions become more accurate and slower. And in this respect, accuracy is positively correlated with reaction time. Okay, so now I'm going to talk about what happens if we consider just one stimulus strength and just one threshold choice. So we're going to hold those other two things constant and just compare the timing of the error responses versus the correct responses. And these randomly selected examples, it looks like the errors were later. But it turns out if you run thousands and thousands of trials in the simulation, the error and correct reaction time distributions are identical. The shape of those distributions are exactly the same. That's predicted analytically from the properties of a diffusion equation, and it's also easily simulated in a simple toy model like I simulated here. So here I've scaled each of these distributions to a sum of 1 so that you can really see that their shapes overlap. And if you do a cumulative distribution of these, the integral of these curves, it's even easier to see that they lie right on top of each other because it tends to smooth out the sampling noise. Okay, so for any given signal strength and threshold, your error trials and correct trials have the identical reaction time distribution. And for a lot of people, this is not intuitive, but nevertheless, it's very well established. So what that means, and just showing those distributions again, and here I've got just the raw frequency distribution, so you can see the number of trials that were correct was more than the number of trials that had errors, and so the animal was getting these, or the model was getting these correct more than chance. But the ratio between these two curves is the same, and therefore if you were to look at the accuracy as a function of the reaction time, it should be flat. It should be independent of reaction time. So this is the third property of the drift diffusion model. Errors and correct responses have the identical reaction time distribution, and therefore accuracy is not correlated with reaction time in this sense. All right, so to sum this up, because signal affects both accuracy and reaction time, as signal strength increases, accuracy increases, reaction time decreases, and so they're anti-correlated. And we attribute this effect to the drift rate. Stronger stimuli have a stronger drift rate in that bias random walk. This is found in all of the extensive literature in the human data and monkey data, and we also see this in our rat data. So this is not a problem. The second property is that as your threshold or criterion increases, decisions become more accurate and slower, and this positively correlates accuracy with reaction time. And this effect is attributed to the threshold of your decision. The more conservative you are, increases both your accuracy and reaction time. And this trend is seen in human data, is seen in monkey data. There are not as much rat data on this, but so far the rat data also agree with this. So this is also not a problem. And the third property is that the drift diffusion model predicts that correct and error responses should have identical reaction time distributions, and therefore accuracy should be independent of reaction time. And this is the problem, okay, that I want to talk about today. It was a lot to take you through, but inevitably, otherwise, halfway through the talk, somebody's like, isn't it just because the stimulus is stronger? Or wait a minute, didn't it just change the threshold? So the problem we have is this one, and it's a problem because it's never seen in real data. So even though this is the dominant model for decision making at this time, accuracy declines with reaction time for monkeys and humans, and it increases in reaction time for rats. So that's why it's of theoretical interest whether either of these facts are true. And if we want to explain decision making, it'd be good if we could explain both of them in a common framework. Great. So that sums up my little review of the drift diffusion model. Now I'll go quickly into some new data. So I showed you data already on these points. I'd like to convince you that you should be suspicious of it, and then I'd like to clear those suspicions. So the most suspicious data would be the rat data because they're completely new and they've only been done once in one lab. We did those experiments using the Ratrix, which is a behavioral setup that was built in my lab and published in 2011. So it's a completely novel set of control software, and everything about it is a completely new implementation. And we implemented our trials in the following way. When the animal initiates a trial, the stimulus pops up instantly, and it persists until the animal makes a decision. Whenever the animal makes a decision, we call that the reaction time, and then the reinforcements happen without any delay. Immediately, they'll either get a drop of water or some timeout. And as soon as the water is delivered or the timeout is over, they are immediately eligible to start another trial. Okay, all these details turn out to be completely different from the human and primate literature. Right? In all the previous literature, there's lots of delays built in, partly to separate different events for the purpose of neurophysiological studies, and partly strategies for training monkeys. You know, monkey person looks at this and says, oh, you can't do this with a monkey because if he knows he's going to get the water the second he responds, he'll be impulsive and he'll decide right away. It's because he's going to have to wait for the water after he makes a decision that encourages the monkey to not make impulsive decisions and so forth. And so the first suspicion really was that we got a different result because we did our experiment a different way. And so we need to test humans and rats on the identical task. Since this is the task we have in my lab, we tested humans on our task. Exact same software. Every line of code that was used to train the humans and the rats is the same. We had to make a couple of changes. First of all, humans won't work for drops of water, so instead we give them a penny every time they get a correct response. So in a 2-hour session, a rat's going to earn 10 mils of water, and in a 2-hour session, undergraduate's going to earn $10 or so if they do a couple thousand trials with reasonable accuracy. The timeouts are the same. Also, we don't want them licking the water ports, and so instead of entering their responses with their tongue like a rat, they use a keyboard. And then another difference, obviously rats' vision isn't the same as humans' visions. Rat vision has a much lower acuity and so forth. So we had to customize the visual stimulus properties to make sure they were in the sensitive range for each animal. Without going into details, I'm showing you psychometric curves from a couple of rats and a couple of humans, just to convince you that we are using stimuli that span the range from undetectable or chance up to ceiling or 100% performance for both the humans and the rats, even though those are different stimulus conditions in the two cases. Okay, so here's an example rat and an example human that are representative. And these are experiments in which we just used one coherence for all the trials. And this is great because we can really sample the reaction time distribution very well in an experiment because we don't have to divide it up into separate analyses for every different coherence that we try. And so you can see here that for the rats, the reaction time distribution of correct trials is definitely shifted to the right. Even easier to see in the cumulative distribution here that the green is to the right of red. And therefore, if you look at accuracy as a function of reaction time, you get a positive slope. And we're going to use the slope of a linear regression through these points as a measure of this effect. And then here's an example human subject in our study under identical task conditions with the caveats I just noted. Here, there's a longer tail in the error distributions and easier to see in the cumulative that the red is to the right of the green, and therefore the accuracy is going down with reaction time. So this is the result that everybody else reports in humans, and we can replicate it under our identical task conditions. And just to remind you, if the drift diffusion model in its elementary form were challenged with the same experiment, we would expect these to lie on top of each other and give no trend with reaction time. Okay, most of the experiments in the previous literature also use psychometric experiments with interleaved different from easy to hard visual stimuli or strong to weak signal. We did that too. So these are mixed coherence experiments, with rats and with humans, showing that except when you're up in the ceiling, for these, every color is a different coherence, and within any one stimulus strength or coherence, it's noisy, but there's a tendency for the accuracy to increase with reaction time. If we just take those stimulus strengths that were in the sensitive range or near the threshold of that animal's behavioral sensitivity, so around 80% correct, between 70 and 90, I think, is the range we took, and then average those together, we can get the accuracy as a function of reaction time sort of summarized over this data set, and we can get a slope. So we're still seeing that for rats, there's a positive slope or an increase in accuracy with reaction time, and for humans, there's a negative slope or a decrease. And these are populations studies, so these are 71 separate experiments on 13 different rats, 71 different experiments on 50 different humans. This task is not fun, so it's hard to get humans to do it more than a couple of times. And this is the distribution of slopes that we got. So positive numbers means a positive slope or an increase in accuracy, and the rat data experimentally almost all falling to the right of 0, whereas the human data are almost all falling to the left of 0 or negative slopes. And this is scatter plots like I showed you before. Either the reaction time of correct trials versus errors, where the rats are falling below the curve, whereas the humans are falling above the curve, like the data I showed you on my conundrum slide. I just have different symbols for each separate coherence analyzed individually. So the message from all of this is we've collected a ton of data. We think that the result is repeatable in rats, and that we are still able to replicate the data that everybody else gets in humans. So we believe rats' accuracy increases with reaction time, whereas humans decrease, and we no longer think it could be attributable to task differences. But is there any way we could still be fooled? So here's a digression to explain why all results of this kind are much harder to establish than I think people realize. So to explain that, I'm going to take a moment to talk about this. Is a painting, a portrait of a pre-Socratic philosopher who was famous for having said, "You can never set foot in the same river twice." So his name is Heraclitus, and this is a Dutch painting from the 1600s. And this character, or this historical philosopher, is always depicted as weeping and wringing his hands. And the reason he's weeping is because he's realized that everything is constantly changing, and he fears that therefore it's impossible to ever arrive at truth. Because no sooner have you discovered some fact about a thing than it's no longer the same thing. And so he's despairing of the possibility of ever knowing anything. And if he ever looked at behavioral data, he would be weeping much more, right? So to get the shape of a reaction time distribution, and especially to clarify what's happening in that long tail, I need hundreds and hundreds of trials, right? It's a very broad distribution. And I need to compare the correct trials to the error trials. So if this subject is 90% correct, I need hundreds and hundreds of error trials, which means I need thousands and thousands of trials of data to establish one of those, slopes that I talked about. And what are the chances that you could study a human or a rat for 1,000 trials on a task and assume that you're still studying the same subject at the end of that time as you were in the beginning of that time, right? Of course, we do obvious things. We wait until they finished learning the task and that their performance appears to be asymptotic. But nevertheless, they are probably changed by the experience of doing the task. And even if they weren't changed by that, they probably just changed by the passage of time. And that's not a huge problem for many kinds of analyses. So if you're just trying to get, let's say, the psychophysical threshold, that change could introduce a little bit of noise in your estimate. But it's a huge problem for this particular arcane detail that we're studying here, and I'm going to explain why. So going back to our toy model, if we imagine that a subject was carrying along with some low decision threshold, let's say for the first 1,000 trials of an experiment, so their reaction times would tend to be short because they are setting a very low criterion for reaching a decision. Their reaction time distributions, if this was a pure drift diffusion model, would have the identical shape and just be scaled. And so their accuracy would be constant as a function of time. If you overlaid the probability distributions or the cumulative probability distributions, it would look exactly like predicted by the drift diffusion model because this was simulated with the drift diffusion model, right? But what if halfway through that subject decided, I'm not getting enough reward here, and they decided to change their strategy and increase their threshold and be much more stringent. And just to make this example really clear, I made a dramatic change. So they've, instead of having 2 units of evidence to make a decision, they're now going to require 5. So what happens is, right, they take much more time on average to reach a decision. The trajectories get much longer, and therefore the reaction time distributions get stretched way out. There's a lot more long reaction time distributions, but it's still the case that if you scaled them, these to probability distributions, they lie exactly on top of each other. And so the accuracy still does not depend on reaction time. So all of this would be consistent with the drift diffusion model. But suppose I didn't know that they changed strategy. Suppose there was a covert internal action of which I did not observe, right? Then I would have just taken all these data from the low threshold, which are perfectly well behaved, and all these data from the high threshold, which are also perfectly well behaved, and mix them all together and analyze them. This is the reaction time distribution I would have seen, right? And it doesn't look obviously funky or too peaked or anything like that. Within noise, it looks like a perfectly good reaction time distribution. And the cumulative distribution here looks just exactly like what I see in rats, right? The correct trials being longer. And that's because changing threshold is the thing that positively correlates accuracy and reaction time. So if I had the, in terms of trial counts, the total number of trials that were correct and error, and I calculated the accuracy as a function of reaction time, I'd see an upward slope. And it would be bullshit, right? And as you can imagine, other manipulations, such as a change, let's say in attention, that might change the rate of accumulation of sensory evidence, could create the opposite effect in a human data set. So this is why you should be really skeptical. And of course, everybody is. And so when you collect the data set, before you analyze it, you usually check to make sure it's stationary, meaning that the data set appears that even though the trial by trial data is very noisy, they seem to be drawn from the same distribution over the entire time of the experiment. And the quick summary of the next two slides is it's really hard to exclude non-stationarity. Because it's, when you have really broad distributions like this, we have a binomial distribution for accuracy. We have these really wide non-Gaussian distributions for reaction times. Your statistical power to tell that those distributions have changed is weak if you only have 100 or 1,000 trials. So you can run a stationarity test and fail to reject the null hypothesis of that it's stationary, and it doesn't mean that it is stationary.
It seems part of the explanation here is that that the rats are more non-stationary than humans.
Well, you might think so, but that's not where we're going to land.
Oh, okay.
So yeah, so that you could say, oh, that's the difference, and that's what all the monkey people said right away. They said, well, that's because rats are dumb and they're bad subjects and they're just more unreliable. Yeah, so let's hold out hope for that hypothesis. Okay, so what do you do when you have data? One thing that we might do is plot out the time series. If I plotted accuracy, it'd just be zeros and ones. It would be hard to visualize. So we just took the average accuracy in, I don't know, a rolling 10 trial bin or something. And that's the blue curve here. So it's fluctuating all over the place. The average reaction time in, I think it's 10 trial window, is these red points. We don't use those smooth points for math. We just show them for visualization. We just do a linear regression from the beginning to the end of the experiment. So this is something like 800 trials, and we just ask, is there a net trend? Is this subject, for example, getting better with practice or getting better with learning, or maybe they're getting tired and it's going downhill? And of course, if you do a regression, you get some slope. You can use something like a Pearson correlation to get a p-value and say, well, those slopes are non-significant. You can also try to check if there's temporal correlations in accuracy. If it's really a stationary process and every trial is independent of the next, right, then the accuracy of one trial should not predict the accuracy of the trials before or after it. So you can just do an autocorrelation of the accuracy. So this peak at 0 offset means that every trial's accuracy is perfectly correlated with itself. And it falls immediately pretty close to 0 for trials that are 1, 2, 3 out to 128 trials away. So this is what you would like to see for something that doesn't have temporal drift or temporal correlation in the behavior. And the red is the same thing for reaction time. So this all looks pretty good. And if we still weren't comfortable, we would shuffle the data. So if you take a time series and you randomize the order of the samples, right, you turn it into a stationary process. Like by definition, those points are now all being drawn from the same distribution. So that's what a truly stationary process should look like. And here's an example of a shuffled data set of this data set below. Again, you get a slope, but it's not significant. The autocorrelations look fine. So this is how we tell ourselves, great, we have stationary data we can analyze. But if you look at the time series, and you smooth it over a wider window, like maybe this is a 50 millisecond running smooth average, accuracy is kind of meandering up and down, the reaction time is meandering up and down. And we scratched our heads and said, I don't know, is this amount of fluctuation what we would expect on chance, or just because we're sampling from these really broad distributions and just by chance we get low or high samples at different times? Or is this subject really drifting around in their behavior? So to look at that, we blocked the data into blocks of 16 trials. And we just got the average accuracy and the average reaction time in each block. These are non-overlapping blocks. And then we also did the autocorrelation of those blocks. So we're looking over slower timescales. And we got the autocorrelations from 1,000 random shuffles of the data, which give you sort of confidence intervals on how much the autocorrelation could be by chance. And here, for this particular experiment, the accuracy autocorrelation looks pretty good out to long term. The reaction time distribution, the autocorrelation, rarely or never exceeds the threshold. Actually, it exceeds the threshold once, but with multiple comparison correction, that's non-significant. At no delay is it significantly more correlated over time than you would expect on chance, but it's above the zero line the whole time, right? So that means there's some slow correlations going on here. If we forget about the temporal order of these 16 trial bins and we just ask, well, what's the distribution of values that we got? So this is just the distribution of reaction times in 16 millisecond bins. And I'm going to focus on reaction times now. It's not Gaussian, but it's some distribution. And we're going to ask, should we have expected that broad of a distribution just from sampling by the simple process of just doing a control calculation where we shuffle the trial order before we break them into blocks. So that's going to tell us how much we should have expected to vary by chance. And what we see here, here brown is the distribution that we got in shuffle data, and blue is the distribution that we got in real data. And an epoch is what we call an experiment that we thought was stationary. Okay, so these are a bunch of epochs from rats, and with the accuracy it's pretty good, but with reaction time it's clearly not, right? The blue, which is the amount of variability in reaction time that we saw in 16 trial blocks, is definitely more than we would have expected in the shuffled control, which is shown in brown, and this is the human data, and it's just as bad. Okay, so in both cases, there's something happening, changing in the animal on some intermediate time scale, right? It's not like if we saw a time series where they were 70% correct and then all of a sudden, bam, they were 90% correct, we'd catch that, right? But the problem is this is drifting in some kind of intermediate time scale that we would never be able to statistically detect. But here's another way of showing that. We just took every symbol here now as one experiment, and I'm again going to focus on the reaction time data here. The reaction time of shuffle control versus the reaction time of the real data, the variance over all the 16 trial blocks. So a lot of variance means that it's fluctuating a lot, and if it's above the line, it means the real data are fluctuating more than you would expect just from sampling of those distributions on chance. And the red symbols are ones that are within experiment, more variable than 99% of the random shuffled controls, so significant with an experiment. So what you see is that almost all the points are above the line, almost all the points are red. It's just as bad for rats as for humans, so they're not stationary. And again, for many analyses, this wouldn't be that big of a deal, but for this really subtle, nerdy point that we're trying to tack down, whether accuracy depends on reaction time, it could completely screw us up. So our solution to that was to come up with a temporally local analysis. And I'm not going to have time to go through the algorithm, but it's really simple conceptually. We just take every error trial that happened. We find the temporally closest trial of the same coherence that was correct. And we get the difference between the reaction time of that correct trial and that error trial. Okay, they're both being drawn from hugely broad distributions, but if they're both being drawn from the same distribution, then on average that difference should be 0. Okay, so we can do that, and that way every time, even if the animal's state is kind of drifting in and out over the course of the experiment, we're only ever comparing a trial to another trial that happened within a couple seconds And so we should be immune to that. And skipping over that didactic to get you to the bottom line, both of the results that I told you could be replicated using this temporally local analysis. So we have coherence of the motion stimulus and the difference between correct and error times. For rats, it's above 0, which means correct trials were longer. This is one rat. This is population summary. This is one human. They're below 0. So the correct trials were faster than the errors. Population summary. And all of these experimental data are available already on bioRxiv, and this was the work of Carly Shabinsky when she was in my lab. And this is just sort of fun that after we went through all the trouble to devise that measure, we said, well, what if we threw at it grossly, totally non-stationary data? We took the lifetime trial of a subject from the very first one they ever did, incorporating all their learning, different experimental conditions, different reward contingencies, everything, and just threw it into a big pot and applied the analysis, and we get exactly the same result, which is sweet because it means we don't have to worry anymore about non-stationarity nearly so much to find out whether or not we're getting this qualitative result for either the rats or the humans. Okay, so the summary of the data part is that this is not attributable to undetected non-stationarity. And I'm going to rush on now to the modeling section. So people have been trying to explain why the primates' accuracy declines with reaction time for a couple of decades, because it wasn't predicted by the drift diffusion model. And there's lots of models out there that can explain it, right? And so one of the ones that I liked the best was the decaying bound model that said that we don't have a decision criterion that's just stable for the whole time of a trial. But once a trial gets going, the longer we've gone without making a decision, the less picky we get about our evidence. And so because the longer time elapses, the less evidence we're requiring, the late decisions are made with poorer evidence, and they're going to have more errors. And this is easy to rationalize. It turns out to be formally equivalent to assuming that there's an urgency signal that's driving them to make a response. And similarly predicted by Bayesian optimality models. But there's other models, including ones that have nothing to do with drift diffusion, like Xiaoxing Wang has a recurrent neural circuit reverberation model that also produces this behavior. So the question is, great, these all explain why we get the surprising late errors that primates show, but can any of them also explain rats? Because I believe there's got to be an underlying conserved mechanism for decision making. And one of the only ones I know of that definitely could explain both is a version of the drift diffusion model. So this is the version I showed you before, where we have time elapsing. I'm just defining some variables that we won't have time to get into now, but you start with some initial position, which doesn't have to be neutral. Then you accumulate evidence at some rate, and then you hit a decision threshold to make a choice. And the extra sauce that makes this model work is that we assume that these variables are not absolutely fixed, but have some noise. So there's noise in exactly where your starting point is at the beginning of a trial. There's a little bit of noise in how fast the evidence accumulates, and also some noise in how this non-decision time, which is all the other events in time that are not part of the accumulation process. So we used this hierarchical drift diffusion model package. I'm sorry you can't quite read that here, but this is a published open source package. It's really cool. That allows us to start with a guess about what all these parameters should be, and then learn by gradient descent a prior for the human species, a separate prior for the rat species, and then individual parameters for every individual in our study. And what the objective function is to minimize the difference between the analytical function's predicted reaction time distribution. So this is showing correct trial reaction time distribution as positive numbers and error trials as negative numbers, just so we can show them all like on one axis there. Red are the actual data from some experiment. Blue is the curve that's produced by the analytical diffusion model. And you just tweak the parameters by gradient descent until you minimize the difference between these and see what you get. And what you get is, here's an example of human data. Here's an example of simulated rat. I'm sorry, this is rat data. This is a simulated rat data. So we fit the model, and then we use the model to generate fake data. And the model is successful at recapitulating the qualitative properties of the rats having positive slopes, correct trials to the right of error trials, and this local reaction time difference being above 0. So it's able to replicate those facts about the rat. And it's also able to replicate the opposite facts of the human, that they have a decreasing accuracy with reaction time. The errors are to the right of the correct trials, and these local time differences below 0, and other details of the data are well recapitulated. And so these, of course, we have to check that these models converge, and they converge consistently and all of that stuff. And so here's what we get out when we look at the model's parameters. So the model parameters say, I'm going to focus on the differences that are the most important. So humans have a higher threshold than rats. So they're more choosy about their distinction, although it's a pretty broad distribution and they overlap. The non-decision time turns out not to matter, so I'm going to skip over that. Humans have better vision, so they have a much stronger drift rate for a given coherence, but we can compare the behavior for different coherences that have the same drift rate, and we still see the difference between the two species. So that's not a surprise. It looks like the rats on the whole have more variability in their starting point of their decision. So their initial, let's say prior about which is going to be the correct response before the trial even begins. Whereas humans have much, much more variability in the drift rate of their diffusion process. So at what rate does evidence accumulate in that given trial? So it's not that rats are more variable and humans are less variable. It's that rats are variable in one way. They have more, let's say, preconceptions before the stimulus begins about what the answer is going to be, whereas humans are variable in a different way. Which is somehow influencing the rate at which the sensory evidence can accumulate. And again, I'm not really a human psychophysics lab, so I'm going to count on the primate people to follow this up, but I'm thinking that it might have something to do more with the sensory end of the coding and attentional mechanisms. But so just for those of you who have to leave, just to summarize the punchline of the modeling part, humans have, so sigma z is the variability in your starting point. Humans have very low variability in their starting point, especially relative to their thresholds, and very high variability in their drift rate. And rats are the opposite, high variability in starting point and low variability in drift rate. And it turns out that if you just do a parameter sweep of this whole parameter space of this model, these are the features that produce the human-like deviation and the rat-like deviation from the original prediction. So here's a model, a stochastic hierarchical drift diffusion model that could explain both rats and humans. I bet there are other models out there. So to facilitate directly comparing them, as soon as this paper is in press somewhere, these data are going to be posted with it so that hopefully other people can find out if their models can also do that. And maybe a take-home lesson that rats are different, have different but not more behavioral variability. And those of you who have to leave, go ahead. I'll just wrap up with a philosophic point. You can tell I'm getting philosophical because the slide's not white anymore. So this is a different actual painting from the Spanish school of Heraclitus, the weeping philosopher. So we wanted to get away from the problem of things constantly changing. And the answer to our simulation was that there's variability, and that explains everything. So we're kind of still back to that point of, oh no, so they're constantly changing. And I just want to point out in terms of our future direction that this variability doesn't have to be noise. It could be reducible to some deterministic factor. And in the rat data, which is what I'll be focusing on, we already have pretty good evidence that there's some role of overall response bias, that a rat just prefers the left response to the right response, can explain some of this, because then in some trials he happens to be biased in the correct direction, and in others he happens to be biased in the wrong direction, and so that's variability in his bias. But even if you get rid of that, we don't make the effect go away. And I think the rest of it is probably, or a lot of the rest of that variability is going to be explainable in terms of the recent history. Where did he get his last reward, and where did he execute his last behavior? And we're working on that. I think the evidence is pretty strong that it's going on, but it's still a little hard to encapsulate. And we imagine that these things are fluctuating on reasonably short timescales because of the timescale analysis that I showed you before. Oops. Right. So this is just a plug for comparative studies and saying that, you know, all kinds of species have to forage persimmons. And I imagine that sensory decision making is probably a very basic phenomenon, and that it would be good if we tried to do very well-matched experiments in different organisms so that we can try and compare them on equal footing. And just conclude with credits. All of the human experimental data were Carly Shavinsky's project in the lab. All of the modeling work was done by Quinn Nguyen in the lab. A whole slew of undergrads were responsible for taking care of the rats during their all of the rat data collection. And we were funded by the Kavli Institute of Brain and Mind at UCSD for this study. Thank you for your attention. Okay, yeah.
So my question is, so you kind of can fit these two different models for the rats and for the humans, and it fits, and you can also interpret what these different parameter settings mean. But now coming back more to a behavioral context, I mean, to some extent, these different parameter settings, maybe you can think about different priors that these animals have in their heads about. So is there some optimalities, or is it, I mean, could one somehow say what the different priors are in the rats and why this maybe be more beneficial for them to take more time?
Right. Okay.
I'm just
right. So
is there an interpretation behind this in terms of behavior, really, of optimal behavior?
Right. In terms of what's differentially optimal. Right. So so now I'm really going to be waving my hands, but right. So the rats, it looks like they're more influenced by somewhat stable or short-lived. Either a stable prior about, let's say, which response is more rewarding, or a short-term history about what has recently been the most rewarding behavior. And that might be adaptive ordinarily in the wild, because in the wild, we don't have 2AFC tasks that are set up in which the contingencies are perversely randomized, right? I've made it so that where you got reward in the last 3 trials has no predictive value about where the next one's going to be. But in the real world, that's probably never the case, right? That's probably good information. So the rats are doing probably the rational thing, and they're not able to override it. Whereas a human subject may come in, and because they're a science undergrad or whatever, they have a prior, it's probably a coin flip. So if I just went right 3 times in a row, if anything, they might be biased to go the other way because they want to even it out, because people are bad with statistics. They know they're in a lab. Yeah. And so that might be one reason for it. Where the variability is coming from with the human case, and again, I'm on thinner ice because it's really not in my area of expertise, but it seems to be, if it's in the rate of accumulation of evidence, it seems like things that would change the baseline firing rate of the sensory neurons, or their excitability, or their correlation of the population, things that can be modified by attention, would make the evidence accumulate faster or slower. And so maybe if there's some top-down attentional modulation that they're trying to figure out the task and see if they can come up with a better way of solving it or something, that might be going on, whether that's adaptive or not. I mean, humans, we're blessed and cursed with cognition and free will, so it may not be adaptive. So I think the rats are doing the rational thing for the real world, if that helps. Actually, you had your hand up.
Yeah, I had a couple of questions. One was about confidence, or some knowledge, right? If I know that I'm not, one other explanation is, if I'm a rat, if I know that I'm really not going to get this, my probability of getting this correct is low, I'm just going to act fast and go to the next trial.
Right. So, and that's what the Bayesian and PMDP models take off on. And I actually don't know if those models are what they're going to do, because they explain the human and rat data fine. Like, the longer I haven't reached a decision, the more I'm beginning to think this is a crappy signal. And therefore, I'm not going to get it right even if I wait forever, so I might as well just guess now, right? In some of those models, I think, hinge on the fact that they're interleaved different stimulus levels. So if they cut bait on the current trial, they're going to get another trial that has a high probability of being better, right? Since both the human and the rat results are found even when we have constant stimulus strength through the whole experiment, that trade-off, that opportunity cost trade-off isn't happening. But right, so for that data, if you could fit the model to the rat data, it seems like it would fit that the longer the rat goes, the more confident they are or something, which I just can't see the rationalization for the opposite result.
If you know that you're going to get another chance, maybe, and if there's enough noise in your signal, and even if it's constant stimulus, you might say, forget about it. You know, if I'm near a
Yeah,
threshold or
yeah,
something,
I-
I'm not going to bet on this.
Yeah,
I'm gonna bet
I-
on the next game where I have better data.
Yeah, so I can't see how that model is going to fit, but it very well might, which is why, because I cannot test all the models, I'm just going to put the data out there. I hope that somebody runs a confidence model. I really hope somebody does the reverberating circuit models and so forth, because I'm not wedded to this is the only way to interpret the data. But what appealed to me about it was that it actually led to some hypotheses of where to look in the behavior or possibly in the brain for these sources of variability. And so it has sort of a pragmatic usefulness. But yeah, I'm not ruling out that any of these other, I don't see how decaying bounds is going to get us anywhere. Because you'll fit that model and you'll find the bounds are getting more and more separated in time, which seems hard to rationalize. But yeah, I think there's a lot of creative work that has gone into the explaining the human and monkey data. and it may very well be that one of the other models will explain the data even better.
So you mentioned briefly, but I'm interested in the sort of quantitative estimate. How much of the variability in the rat's behavior could you predict if you looked at his bias, let's say assessed on zero coherence trials in some little interval? How much of that explains the behavior in N trials around that? That cluster of points you have, above zero. In the
Yeah, so one reason I didn't show that is for time, but mostly is because we're still trying to come up with the best way to distill that in a clear metric. We can actually just do a information theoretical analysis of how much information does it contain, or I do know if we take only experiments in which there was no overt bias, we still get the same results. But if we fit the model where, like, say, all the leftward trials get their own parameters and all the rightward trials get their own parameters, right? Then we do get a model that fits the data better, even after compensating for the fact that it has more parameters. But if we take into account the previous trial's location and outcome, we explain the data much better still. So we're still just, and I don't know if we have enough statistical power to really determine the dependency on longer sequences, although we could just average over longer windows. But yeah, so how much of it is a number I haven't nailed down yet. Yeah.
I'm curious if you've been able to get your hands on other modality data from auditory or olfactory, and whether the prior bias in the rodents might be modality specific, and if you rely on priors more, we have low acuity for the perception.
I see. So you're saying because if rats are just less visual and more olfactory, then maybe they would rely more on. So I don't have those data. I've asked around for data, although more I've looked for people who have visual data like this from other species. It's always hard to extract data, as those of you who are theorists know. So I'm hoping that maybe if I deposit some data, then maybe other people will deposit data too. But I will say that one thing that's kind of cool in the data from the rats is many rats exhibit a bias only on low coherence stimuli. So as the weaker the signal to noise ratio in the stimulus, the more they rely on their bias to make their decision. So they can be 100% correct for a high contrast stimuli, but their errors that they make on low coherence stimuli are increasingly explained by the bias. So that's sort of not the same thing you're asking, but yeah. And I think the thing about comparative studies is it's hard work. We were talking about this before. Animal behavior is not as easy as I think some people just assume it is. It's like, oh, well, I'll just throw some animals and I'll do a test and get some data. We spend a lot of time trying to get inside the head of rats and understand how to train them and how to interpret their data and what to be cautious about and so forth. I think it'd be foolish for me to set up a fruit fly experiment, right? A fruit fly person should do that because they're going to be better qualified than I am. And so I think requires more of a consortium approach of people who are experts in different sensory modalities or in different species, agreeing to all do the same experiment or a formally comparable experiment. The data that we get for this vision experiment in rats, we get the same thing in mice. And we didn't do random dots in mice, but we did other visual stimuli. We get the same thing for all different kinds of other visual stimuli in rats. So it doesn't have to be random dots. So that holds out hope that you can find an experiment in audition or olfaction that's somehow formally equivalent.
Yeah. Along those lines, I guess mean, one of the striking differences between the human and rat was in the drift rate.
Mhm.
You know, so that sort of suggests that the signal for this particular task was maybe very weak for the rat. You know, and if we could find another
Right.
task that had a stronger sort of comparable signal, and like a similar kind of drift rate,
Right.
right? Then it might be more of an apples for apples comparison. Because it maybe is something about just being in this regime where it's kind of like something that the rat is not struggling with, you know, in a way that a human isn't. That might
Yeah.
contribute to.
So fair point. It could be. It was a harder task for the rats than it was for the humans.
Mm-hmm.
We can pick out the coherence that was equally hard, you know, and compare them. It was true, and I skipped over this detail for time, but in both the humans and the rats, their deviation from the basic drift diffusion model became stronger and stronger the stronger the stimulus was.
Oh.
So high coherence stimuli had the biggest, for the highest coherence stimuli, the humans had the most late errors, and the rats had the most early
I
errors.
see.
So it seems like if we were to keep getting stronger stimuli for the rats, they might just continue to diverge more in that direction. But yeah.
I have a modeling question.
Yeah.
So I've worked with more of the vanilla drift
Right.
diffusion model with just the 4 parameters
Yep.
and the variance. Have you tried applying that more vanilla version without the bells and whistles of the rat like?
Yes, absolutely. Here it is. So yeah, if you take the vanilla model, you can get the psychometric curve just fine, but you get the reaction time distributions of correct and error trials sitting right on top of each other. They're identical. You get accuracy being completely independent of reaction time. And these different temporal time differences between the correct and error trials just hover around 0. So yeah, sure. Yeah?
I like the idea that the start point of rats, humans is it's more variable in rats. But I'm not sure if I missed something, or if it's possible that so some kind of general arousal state could that is time varying rather over the whole trial rather than just the start point might be important here.
Right.
Did you rule out the possibility that there might be something, maybe there's a decision point, a random decision time that, the rat just simply decides, I'll make a decision based on how far drifted it is, rather than a threshold?
Right. So before we did this model, so one of the ways you could explain the rat data is if the rat just had a deadline, but it's just a noisy deadline. And whenever the deadline came, he just decided on however much evidence he happened to have. Right. And that would qualitatively produce that same pattern. I don't know if you would get the shape of the reaction time distributions, this skewed shape that you get. So I haven't ruled that out. I haven't really ruled anything out. I've just shown that it's possible to account for the data with a drift diffusion model without a lot of extra ad hoc bags being thrown on the side of it. But it's not really satisfying until we pin down what the variability is. And I didn't explore at all variability of things changing over the course of a trial. Definitely not ruled out. Okay.
Thanks, Pam.
Thank you.
Great. For those who